Welcome to our webinar, Microbiome Research Streamlined with Phenotypic Characterization using PreBioM Plates. My name is Kasia Proctor, and I will be your moderator today. First, I have a couple of housekeeping items I wanted to share. This webinar is being recorded, and I will be sending out a link to the recording to everyone that has registered. If you have questions, please submit them in the Q&A section. There's a little chat window below with a Q&A, and you can submit them there. Many of you have submitted questions during the registration. We appreciate those, and I have shared them with our presenter. They will be, if we don't get to them during the presentation, we will try to get to as many of the questions as we can at the end. Okay, now let me introduce our speaker. Juan Sanchez is a product manager here at Biolog. As a former Stanford scientist, he co-authored several gut microbiome publications featured in Nature. Juan, please take it away.
Thank you, Kasia. Good morning, everyone. I'm Juan Sanchez, the Consumables Product Manager at Biolog. Today, I'm thrilled to demonstrate how the PreBioM plate line combined with our Odin instrument can help you unlock deeper insights for your microbiome samples. But first, did you know prebiotic consumption has been linked to longevity of centurions? Prebiotics like inulin present in the PreBioM plate line enable the improved colonization of bifidobacteria, which assist in regulating metabolic function, immune response, and inflammation. This serves as just one more reason to study the interactions between prebiotics and the gut microbiome, with a lot more exciting discoveries to be made. Today's agenda is as follows. I'll start with an overview of the prebiotics and gut microbiome, including a look at the various omics techniques currently employed by microbiome researchers. Then I'll present our PreBioM plate line and its capabilities. Once we've covered the basics, we'll dive into the main event, a case study demonstrating how PreBioM plates and Odin can be used to compare prebiotic and pathogenic E. coli strains under multiple atmospheres. Finally, I'll summarize our key findings and open the floor to questions. Let's start with prebiotics, which can come from fermented foods like kimchi or be isolated molecules like inulin. These provide energy, metabolic substrates, and essential micronutrients selectively to beneficial bacteria, like those found in probiotic supplements. Prebiotics also happen to be the substrates in our PreBioM plate line. In essence, prebiotics support probiotic microbes present all over the body, including the skin, mouth, and gut, which is special due to its low oxygen content, enabling the growth of obligate anaerobes and facultative anaerobes, like the E. coli strains we will examine later. Once inside the gut, prebiotics and probiotics work together to enable a healthy, balanced gut microbiome. The microbiome has huge impacts on the host, from regulating how drugs are metabolized, similar to an additional liver or another organ, to regulating neurological conditions such as autism by postbiotic molecules generated by the probiotic bacteria, which can travel anywhere in the body. From my point of view, no omics approach is complete without phenomics data, serving as the final output of all of the complexity within the cell. A variety of omics techniques are used to study the gut microbiome, from genomics methods like next-generation sequencing and 16S, to proteomics and metabolomics, which rely largely on NMR, as well as chromatography and mass spectrometry. As we travel up in the chain of the various omics, there are more regulatory mechanisms, meaning that a change in the genes may not correlate with the output, which is the phenotype. Fortunately, at the end of this chain is phenomics and metabolic phenotyping, which serve as the output of all of the regulatory cellular mechanisms and is what biolog assays measure. Our phenomics technology revolves around watching what a microbe consumes and how efficiently that substrate turns into biomass or fuel, and whether that fuel is expressed as respiration or growth. The breakdown of food sources is enzymatically controlled. Enzymes work with cofactors like NADPH to turn over substrates. Our assays use a tetrasodium redox dye to report on these reactions via color change, and we measure the absorbance/production of the dye as shown in the following slide. Here we can see the biolog system in action. It encompasses a 96-volt plate preloaded with relevant substrates such as prebiotics. By analyzing expressed cellular properties or phenotypes, we eliminate the noise and focus on properties that are biologically relevant to the cells of interest. We combine these microplate assays with Odin, a versatile and high-throughput solution for dynamic incubation and measurement of up to 50 plates. Odin is supported by its own intuitive software to compute multiple parameters for kinetic characterization and comparison. This results in more robust experiments with actionable data. Together with PreBioM Plates, Odin provides an excellent solution to study the microbiome, including support for facultative and obligate anaerobes, as well as aerobic microbes, via 3 new protocols available for download at biolog.com. With Odin, kinetic growth and metabolism data can be automatically collected at the scale necessary to test profound scientific questions. Additionally, Odin's features and streamlined reports facilitate community analysis, which is of great importance to microbiome research. Next, I introduce the PreBioM plate line. The PreBioM plate line is a set of 3 microbial phenotyping microplates packed with prebiotic substrates. These phenotyping plates cover a menu of prebiotic substrates increasing in complexity from simple sugars to macromolecules and can be used independently or as a set. The plates enable the utilization of built-in triplicates as pictured here, enabling statistical analysis downstream. The first plate, PreBioM 1, contains monosaccharides such as glucose and fructose, pictured in yellow, as well as disaccharides like sucrose, which combine 2 monosaccharides via a glycosidic bond. Due to their simple structure, bacteria can readily absorb and metabolize them, providing a rapid energy source. The PreBioM 2 plate contains oligo and polysaccharides, which are utilized a bit differently due to their structural complexity. Bacteria often need to enzymatically break down polysaccharides into monosaccharides before they can be utilized for energy, making this process slower, but in exchange, it can provide a more sustained energy source. Raffinose, an oligosaccharide, is composed of 3 sugars, fructose, glucose, and galactose, while inulin, a polysaccharide, is composed of only 2 sugars, glucose and repeating fructose units, like a polymer. As mentioned earlier, inulin has gained a lot of attention in recent years as a prebiotic that promotes the growth of probiotic bacteria and inhibits the growth of pathobionts. Plate 3 contains dietary fibers and food extracts. Many popular commercially available fibers contain dextrin mixtures, which can vary in resistance to metabolism based on branching from alpha-1,6 linkages. In addition to dietary fibers, PreBioM contains prebiotic food extracts like stevia leaf extract, which has a rich mixture of metabolites of various sizes and complexities. PreBioM plates can be leveraged to test interactions between microbiome community members and prebiotics. The plates accommodate diverse aerobic or anaerobic microbes, including facultative and obligate anaerobes relative to the gut microbiome. And they afford an understanding of substrate utilization, leading to the discovery of beneficial symbiotic combinations. As shown by the growth curves, PreBioM plates enable high reproducibility between built-in replicates. The aggregation of replicates and the calculation of plate level parameters and kinetic measurement provide resolution into the lag and log phases, ensuring all aspects of your experiment are captured and the correct conclusions can be reached. Now for our main event, a case study comparing pathogenic and probiotic E. coli strains. For this study, two strains, E. coli Nissl 1917, a probiotic strain, and E. coli O157H7, a Shiga toxigenic pathogenic strain, were selected. These strains are facultative anaerobes, meaning they grow well in aerobic conditions and can adapt to anaerobic conditions like those present in the gut. Multidimensional phenotype profiles were obtained by measuring energy metabolism and growth kinetics under aerobic and anaerobic conditions using the methods described in our new PreBioM protocols. Each of the 3 plate sectors was used as a technical replicate, assisting in downstream data analysis, and 24 plates across all study conditions were simultaneously incubated for 24 hours on an Odin instrument with reads at every 20 minutes. As shown in our comparable genome map, the pathogenic and prebiotic E. coli strains are not too genetically dissimilar, with E. coli Nissl 1917, our probiotic strain, having less than 1,000 unique coding sequences. This means that results we get out of a genomics experiment may not be enough to elucidate the differences between the 2 strains and engineer strategies to promote the probiotic strain while hindering the pathogenic one. An adjusted p-value of less than 0.05 and a log to fold change below -1.5 and above 1.5 were used to identify 15 differentially utilized substrates across the 3 PreBioM plates, with most substrates occurring in the first and second PreBioM plate due to regulation of enzymes. Principal component analysis using the substrates as loading for cell growth reveals atmosphere-dependent class separation on principal component 1, accounting for 64.1% of the variance, and strain-dependent class separation on principal component 2, accounting for 11% of the variance. Similarly, principal component analysis using the substrates as loadings for cell metabolism reveals atmosphere-dependent class separation on principal component 1, accounting for a massive 69.47% of the variance, and strain-dependent class separation on PC2, accounting for 19.62%. This is further supported by the limited overlap in the differential substrate utilization under the 2 atmospheric conditions shown in the following Venn diagrams. For growth, we can see that only 2 overlapping metabolites between anaerobic and aerobic growth were identified, mirroring the importance atmosphere had on the principal components. These substrates also overlap well with the loadings, but contributing to the class separation in the principal components analysis. Similarly, for metabolism, only 2 overlapping substrates of interest were identified, raffinose and sorbitol. Here we examine the cell growth and energy metabolism of cells incubated in the presence of allose, a monosaccharide in the PreBioM plate. E. coli Nissl 1917's utilization of allose for growth under aerobic and anaerobic conditions, pink and orange, underscores the importance of Odin's kinetic measuring capabilities for studying the gut microbiome. Despite reaching equivalent endpoint measurements, the growth rate under anaerobic conditions lags behind that of aerobic conditions. Selecting the wrong endpoint could result in incorrect observations. Additionally, O157H7 showed limited to no growth using allose. There was no energy metabolism for O157H7 in either atmosphere and substantially more energy metabolism for Nissl under aerobic conditions. Area under the curve shows E. coli Nissl 1917 successfully used allose for growth. Additionally, energy metabolism was active for allose under almost all atmospheric conditions. For anaerobic culture conditions, there is almost no anaerobic dye reduction, but there is growth, suggesting an ability to divert utilization into biomass rather than energy metabolism. Allose was inaccessible under all conditions to its pathogenic counterpart, E. coli O157H7. To help explain these findings, searches for allose were conducted for both strains using the BioPsych database, revealing implicated genes and enzymes annotated only for E. coli Nissl 1917. Allose substrate utilization is justified by gene ontology in this case, but this isn't the case for many other substrates, underscoring the importance of phenotypic profiling as a direct measurement of the gene and enzyme output. Here we can see very limited aerobic growth for either strain using inulin as a substrate, but substantial anaerobic growth with E. coli Nissl outperforming O157H7, demonstrating the dominant role played by the atmosphere over the strain. Both strains showed similar aerobic metabolism. However, E. coli O157H7 outperformed E. coli Nissl anaerobically. In this case, inulin was not used substantially aerobically for growth by either strain, and it was used anaerobically for growth significantly more by Nissl 1917 than O157H7, the pathogenic strain. Searching both strains in BioPsych reveals only one annotation present for O157H7, a fructotransferase enzyme, highlighting the disconnect between genomic and phenotypic data. Our results suggest inulin could afford the probiotic strain an advantage over its pathogenic counterpart. Looking at growth by a OD for sucrose, sucrose was exclusively utilized by E. coli O157H7, and this was the case for both growth and energy metabolism, as we can see here. This is despite having the same genetic potential, according to our database. Exclusive support for growth or metabolism was also tied to the culture conditions. Once again, these results highlight the disconnect between gene ontology and phenotyping. Despite having related genes, E. coli Nissl does not appear to use the substrates for growth or energy metabolism. Next, we look at kinetic curves for raffinose. For growth, an endpoint read at 12 hours would tell a very different story and may lead to an incorrect conclusion, as O157H7 in dark blue and Nissl 1917 in pink reach similar OD values after 24 hours under aerobic conditions. Energy metabolism shows similar intricacies, with O157H7 aerobic in dark blue initially lagging, only to reach a similar endpoint to strains cultured anaerobically. Here we see no anaerobic growth for E. coli Nissl 1917, given the substrate raffinose. This is interesting because raffinose energy metabolism is still present under anaerobic conditions and found at similar levels to aerobic metabolism, highlighting the environment's role in making important adaptations for facultative anaerobes. A search on Biopsych reveals both strains share the same annotations, suggesting they should behave similarly. However, E. coli O157H7 has higher levels of growth and energy metabolism than E. coli Nissl 1917, discoverable only via phenotyping. The breadth of substrates in PreBioM plates uncovered a competitive inhibition between fructooligosaccharides, FOS, and glucose. Growth data shows that E. coli O157H7 utilized FOS significantly more than Nissl 1917 in the absence of glucose. Excitingly, a previous publication by Kaplan et al. Corroborates this finding, which leads us to wonder what other exciting discoveries can be made with PreBioM plates. Our omics approach is no longer missing out thanks to phenomics data collection enabled by Odin and PreBioM plates. In summary, significant substrate utilization differences were found between strains and growth conditions, despite having genetic similarities. Atmosphere rather than strain accounted for most variants, which has implications on gene function and enzyme activity, especially for facultative anaerobes, which were grown concurrently under 2 atmospheric conditions thanks to Odin. Effortless kinetic monitoring of cell growth and metabolism showed the substrate's impact on the lag and log phases, giving us an unbiased view of the experiment and helping us reach the correct conclusion. Substrates like allose and sucrose were extensively used by either strain. Substrates like inulin could only be utilized either aerobically or anaerobically by either strain. Despite aerobic growth and metabolism, substrates like raffinose did not support aerobic growth, but retained anaerobic metabolic activity. Our multidimensional approach utilizing PreBioM plates and Odin provided a detailed mechanistic insight into how prebiotic substrates influence bacterial growth and metabolism. The observed variations in substrate utilization underscore the limitations of relying solely on genetic information to predict metabolic phenotype. The findings emphasize the importance of phenotypic profiling for accurately understanding and manipulating microbial interactions within the gut microbiome. As we conclude our exploration of the gut microbiome, I'd like to highlight some additional components of the biolog ecosystem that may pique your interest. Firstly, we offer a comprehensive range of PM phenotypic microplates. These plates are utilized in Odin to analyze substrate utilization and chemical sensitivity. Similar to PreBioM plates, PM plates are designed to support the culture of both facultative and obligate anaerobes and come with their own protocols. For those studying ecological microbiomes, our EcoPlate provides a turnkey solution for phenotyping microbial communities in soil, waste, and water samples. If you need to identify specific microbes, Biolog also provides identification test panels for a wide variety of organisms, including gram-positive and gram-negative bacteria, anaerobic bacteria, yeast, and filamentous fungi. Finally, if multi-top identification, genomic sequencing, enumeration, or phenotyping and Odin are required, our fully certified lab is equipped to support and advance your projects. Thank you for joining this webinar. We will now move on to the questions section.
On. Okay, so as mentioned before, if you have questions, please submit them through the chat Q&A. I see we already have a few questions. Okay. What criteria are used to select the substrates for the PreBioM plate?
Oh, that's a good one. The substrates for the PreBioM plate were selected with the help of key opinion leaders in both industry and academia, and from an extensive review of current trends in the literature to offer a relevant, comprehensive look into the interactions between the microbiome and prebiotics. These substrates, in order to go into the biolog plates, had to have some stability and solubility, so these were additional parameters that we looked into.
What are some of the challenges you encountered where comparing strains under aerobic and anaerobic conditions with the PreBioM plate line?
Well, 24 plates at a time under 2 atmospheric conditions doesn't sound like a very large experiment, but it was. Without Odin, I don't think I could have done this experiment and gotten the quality of data needed to achieve the findings in the presentation. Handling of the plates overall between the two was the most difficult.
Okay. Let's see, what are some other questions? How do the findings from the aerobic versus anaerobic conditions contribute to our understanding of microbial dynamics in the human and animal gut?
Sure, the human and animal gut is predominantly an anaerobic environment, but there are regions such as the upper gastrointestinal tract where oxygen levels are a bit higher. Studies of microbial dynamics under different oxygen conditions can reveal how microbial communities shift between these areas. Aerobic conditions may favor the growth of specific microbes while inhibiting others, and understanding these dynamics helps explain how the microbial community composition varies along the gut and how it might change in response to dietary, environmental, or health factors.
Let's see, there's a question here about dye loading. Does the dye or loading media interfere with downstream analysis? For example, would it be possible to recover the broth after assays for transcriptomics?
I'm not entirely sure about recovering the broth for transcriptomic assays after using the dye, but for example, for metabolomics assays, there can be different techniques applied in order to not have the interference of the dye and analyze those postbiotic molecules, as well as measure how much of the substrates were consumed and what was produced. So people have been able to multiplex them with different omics, such as metabolomics.
Yeah. Sorry, just trying to keep track of the questions here. Do the PreBioM plates contain more complex sources, require longer incubation time than the traditional PM plates?
The PreBioM plates and PM plates are a bit different, with the PreBioM plates being tailored specifically to analyze the microbiome and its interactions with prebiotic substrates. However, both the PM plates and PreBioM plates require similar timings and have different substrates of varying complexity. So overall, I think there are two complementary plates that can be used as a system to really phenotype your entire organism and get an in-depth understanding.
How did you run the plates in anaerobic conditions? Did you seal biolog plate in a transparent bag in an anaerobic cabinet before loading into Odin?
Okay. So in order to run aerobic and anaerobic conditions simultaneously, the plates were sealed using a plate seal within an anaerobic chamber after inoculation and then transferred to Odin for growth for 24 hours. We've analyzed doing this system using the plate seals or doing it in the anaerobic chamber and have found very similar, if not identical, results.
So I want to make sure we get to some of the questions submitted during the registration. There was a question from PreBioM compatibility with one instrument, and what kind of data can, I'm sorry, it's not, it's probably a translation here. So PreBioM compatibility with one instrument and what kind of data can be improved using PreBioM plates? Sounds like they have an application in several research topics.
Yeah. For example, PreBioM plates can be coupled with multiple omics techniques such as metabolomics and proteomics. There are still cells growing within the wells. So in addition to the phenotypic data, mass spectrometric data can be obtained, and other colorimetric assays can also be performed using these. So there is the capability for multiplexing and getting reads from different instruments out of your PreBioM plates. And the insights you're going to get out of this multitude of data sources is that you'll be able to make different predictions. For example, since the data from PreBioM plates comes from in vitro cells, you could tie this to an in vivo analysis from bacteria sourced from feces, for example, in which you can compare levels of different metabolites and see how they performed on the PreBioM plate for substrate utilization.
Question about future development plans for more PreBioM plates with different substrates.
Yeah, absolutely. We're always interested in keeping the PreBioM plate line relevant. We continuously interact with our stakeholders, which is the research community, and finding what substrates they're interested in. And when these are relevant, we can, of course, make new plates and expand this plate line. As a matter of fact, I would be happy to set up a chat if you have some ideas.
Yes, we will be to add to Juan's comment here. We'll be following up after the webinar as well with some of the questions that we might not have time to get to or with specific requests. There's a question about water analysis. I can see that the equipment can be used for water analysis. Can we use for fresh water, marine water, wastewater? Is there any interference present in the analysis with the composition of the water?
For this type of use case, I believe the EcoPlate may be a better solution for analyzing water, including wastewater. I don't expect to see much interference coming from the matrix, but this would, of course, need to be tested out experimentally. There are many publications using it already, and I don't think I've seen interference be a particular impediment here.
Okay. Do you need the Odin to read out the PreBioM plates, or is it compatible with fluorescent reader?
PreBioM plates do not have any fluorescent capabilities, and using them with Odin really facilitates the whole workflow. As I mentioned earlier, one of the biggest challenges from performing a 24-plate study across two different atmospheric conditions was handling all of these plates I could not imagine having to do this with a plate reader, although it is possible. It would be very difficult to manually take these time points for the different plates every 20 minutes. It might take weeks. Additionally, the data output would be somewhat difficult to read, which is somewhere where Odin really shines. It makes getting insights from this prebiotic data much simpler. So although it is possible to do this on a plate reader, I would highly recommend performing it on an Odin. And we have two different models to accommodate either 50 or 8 plates at a time. So there's no one size fits all.
There's a question about dye here. Does the dye work intracellular or extracellular?
Regarding the dye, some of the like the NADPH would be intracellular. But yeah, I think some of that I'm not entirely able to expand on. Sorry.
Should dilutions be made in a specific fluid as for anaerobic plates?
Yes, we do offer a specific fluid, an inoculating fluid to perform the dilutions stated in our protocol. That is IF0A, IF0 anaerobic. And it is available for purchase from Biolog. So yes, there is a fluid available.
Thanks. Can we request a custom plate with substrates from each of the 3 PreBioM plates?
Yes, that is a capability that we currently have. So some of our customers have very specific conditions that they want to test, and running all 3 PreBioM plates may not be ideal. They might want, as a matter of fact, some of the substrates found in our PM plates. So we offer combining these substrates from the different plate lines into custom plates for users. These are manufactured at high quality using our systems and are available for purchase. Please reach out to a field service or a sales person for these.
Okay. There is a separate question. I just want to acknowledge it here because there's somebody's asking to see a report. So we can follow up offline to show that because I know that Juan has his slides pulled up right now. So we'll do that after the webinar. Let's see, some other questions are any benefits for IBS? How specific regimens can be tailored to individuals?
Yeah, absolutely. Various prebiotic substrates have been shown to lessen the impact of IBS, as well as different microbial communities, and the richness of the communities has been known to impact those effects of IBS. So substrates like inulin, for example, may be beneficial in this case, which are present in the PreBioM plates. I'm sure there's more substrates that would be beneficial to this use case in particular, but they're very broad ranging. So it's not tailored to one single medical condition, but you can also target neurological conditions, immunology. The sky is really the limit with these plates.
Okay. Do you also have data you could share using complex microbiome samples compared to single isolates?
Yes, we do have data, and I'm actually working on a new app note for this. So once that is released, I'd be more than happy to share it with you. For the moment, I recommend looking at our other application notes as well as webinars and what is available in the literature to see these communities.
We investigate microalgae for wastewater treatment. Do you have any suggestions how we could use a microplate in this case?
For using microalgae in wastewater treatment, I guess the ecoplate would once again be the solution here since we are dealing with wastewater. And what is different in the ecoplate is the simplicity of this plate. Uh, there is a gel matrix within this plate where the samples are directly loaded. So it's a very simple protocol to follow. And you should be able to get results out of this using that sample type.
Okay. Somebody made a comment that they're interested in a study of an oral microbiome. Do you want to comment on that one?
Great. Yeah, that's a good one. So although this webinar was tailored to the gut microbiome, the substrates on the PreBioM plate line apply to various other microbiomes, such as the oral, vaginal, skin microbiome. We looked at different publications and got these substrates from them. So I know for a fact that the salivary microbiome is well covered in PreBioM plates.
Okay. And we had a couple questions about the timing, so I'll ask this one, but I think this will be our last question, and then anything we did not get to, we'll take offline later. Whilst you recommend a longer incubation period to get the comprehensive kinetic growth and metabolism data, what's the quickest you can currently detect the substrate utilization? For example, in E. coli, are you looking into novel ways to speed it up?
Sure. In this experiment, we incubated for 24 hours, but as the data showed, this might not have been necessary for many of the substrates which were rapidly utilized, particularly the more simple ones. So you are able to get insights at much earlier time points. However, I do recommend waiting until the cell culture has reached a exponential curve and is no longer intensely growing. So to expedite this, what are we looking at? That's still something under development, but should there be a need for quicker, higher throughput, this is of course something we can look at and target.
Okay, great. Thank you. So with that, we will end the Q&A session. Thank you again to everyone who has joined us. As mentioned, we will be sending out the recording. If we did not get to your question, we'll follow up separately, although we did have a lot of questions, so really appreciate the audience participation here. Thank you, Juan, for your presentation, and thank you to our audience for joining, and we will talk to you next time on our next webinar. Goodbye.